Hi there, everybody. This is the Cells Unit Lecture Part 2. I'm going to get into more detail in some of the organelles now. We ended last time with cytoplasm. We are now on number 3 organelle called the nucleus. There's a couple pictures I'm showing you over here of the nucleus just so you can have an idea of what it looks like. This shows you the nucleus. It's also attached to this thing called the endoplasmic reticulum. So this is the nucleus, this dark purple and light purple area right here. Over here, here's another one. The yellow here is showing you all the different parts. There's a lot of details, a lot of labels. Let's just go through them so you can make some sense out of it. What's found inside of the nucleus? Well, DNA and RNA. Your nucleic acids are found inside of the nucleus. This is the information of the cell. This controls how the cell works. That's where it's all found. So we also call it the control center. Inside of here is a jelly-like liquid. It's called the nucleoplasm. It's kind of like the cytoplasm, but the cytoplasm of the nucleus, basically. Jelly-like liquidy area that kind of houses it. All right. Chromatin. This squiggly lines, this is loose DNA. All right. Loose DNA is a chromosome. Excuse me. Loose DNA is called chromatin. When DNA is wound up really tight and you can actually see the physical structures of chromosomes, then we call it a chromosome. So it has to be wound up, condensed really tight, and DNA is not visible as chromatin. We're showing it to you in this picture, but in reality, it would be so thin you would not be able to look at it. You'd not be able to see it under a microscope. You could see chromosomes under a microscope. You cannot see chromatin under a microscope. Imagine this. Chromatin would be like a string, okay? Chromosomes would be like string wrapped around something, and a lot of it wrapped around something. You would see it easier. So chromosomes are visible under a microscope. Chromatin is not. What does this mean? Well, there's this important thing coming up in a later chapter called DNA replication. Well, it's important. DNA has to copy itself, right? And that happens in the nucleus. Transcription is an important process we're going to learn about later also when we get into the DNA chapter. And transcription has to do with the process of making proteins. And those are very important. So replication and transcription occur here inside the nucleus. The outside of the nucleus, this dark purpley area, or this, I guess, like orangish yellow out here, this is the nuclear membrane, also called the nuclear envelope. It's made out of 2 bilayers. So there's a bilayer on the inside and a bilayer on the outside. So one bilayer faces the cytoplasm, one bilayer faces the inside, the nucleoplasm. Okay, so it has a double bilayer here. All right. You have little holes. You have little holes. You can see them here. Little holes, little holes. They're called nuclear pores so that things can enter and leave. They're pretty small. They're only letting certain things like RNA possibly leave. DNA cannot get through there. It's too big. So RNA can pass through these pores, but not DNA. The nucleolus, it's the darker area, Well, what you have here is you have some packed DNA. This DNA is making something there. It's making what's called rRNA, and which are what ribosomes are made out of. So these are the protein factories of the cell. We spoke about these proteins, the importance of proteins in our last chapter. I told you that proteins are important because you're talking about all the enzymes that control the way your body works. So the nucleolus makes the rRNA and basically the ribosomes, rRNA is what ribosomes are made out of, and the ribosomes are your protein factories. So the protein factories are essentially made inside of the nucleus, in a specific area called the nucleolus. Mitochondria, we're going to talk about these in a great deal of a lot of detail later when we discuss how energy is made, but let's go through it now. This is the organelle that creates energy. Energy for you and me that we use, or usable energy, is called ATP. Adenosine triphosphate. So this is usable energy that you and I can use and run our cells processes with. There's 2 parts of and by the way, making ATP, the process of doing that is called cellular respiration. So what happens here? Cellular respiration. What happens here? Energy production happens here. All right. 2 parts occur here. All right. 2 parts of this energy production. One is called the citric acid cycle and the electron transport chain. And they're very important parts. I'm just mentioning them now. We'll go over them in detail in a couple chapters from now. Mitochondria have 2 areas. They have an outer membrane, and then they have this other inner membrane here that winds around. Okay? 2 membranes. All right? They have these folded areas. These folded areas are called cristae. I mentioned to you in the last lecture that increasing surface area is very important. Well, this is our energy producer. We need to increase. It needs to be more efficient. So what does it do? It creates folds in these inner membranes that increase surface area. So now ATP can be produced at a much more rapid rate because you have more space to do things. So creating folds increases surface area in a structure. Mitochondria, they're a little special. They have their own DNA. So this is a unique feature of two organelles. They have their own DNA. They're called mitochondrial DNA. Your mitochondrial DNA in your cells, you received it from your mother. All of your mitochondria that you have in your body, your DNA in that mitochondria is maternal DNA. So if in a crime scene investigation, if they were to discover mitochondrial DNA, they could link it to the person's mother, but not their father. And that's because the egg, which produces the embryo, is the feature that contains the mitochondria in the newborn. So mitochondrial DNA always comes from the egg, which comes from the mom, basically. Also, these guys, they have their own ribosomes. I'm going to mention to you in a second that these are the protein factories, and they have their own proteins called enzymes. So they have their own enzymes which control the reactions that they need done inside of them. Ribosomes, they have two parts. These are your protein factories. Fancy word for making synthesis or synthesizing. They have two parts, a larger part called the large subunit and a smaller part called the small subunit. They come together when they want to make proteins. And you notice this is showing you a growing peptide or a polypeptide chain made out of amino acids, which will eventually become a protein. Okay, so these are your protein factories. They're making these polypeptide chains that become proteins. Okay, what is this made out of? I already mentioned to you earlier. They're made out of this thing called rRNA. Ribosomal RNA is what they're made out of, and they're made out of proteins. Okay, sometimes they're attached to the endoplasmic reticulum. Sometimes they're floating in the cytoplasm. We call them free ribosomes. By the way, they have no membrane around them. So these are found in eukaryotes and prokaryotes. ER, endoplasmic reticulum, there's two types, and we're going to talk about the detail of each type in a second. Well, what are they? They're these membrane-bound compartments. They're like tunnels or a subway system or streets. It's a transportation network for the cell. If you look over here at the picture, ones that have dots attached to them, okay, dots all around them, are called rough ER, and those dots are ribosomes. Some of them don't have dots attached to them, and those are called smooth ER. So if they have dots attached to them, they're rough. No dots attached to them, then they're smooth ER. Okay, so they're made up of these membrane tubes and bags and sacs, and they're called cisternae. That's what they're called. All right, rough ER. Okay, this is rough ER over here. How do we know it's rough? There's ribosomes attached to the surface of it. If it has ribosomes attached to the surface, it looks rough. Therefore, that's why we call it RER, or rough endoplasmic reticulum. What is the job of this area here? Well, the job of the rough ER, it makes a lot of things for the cell. Well, since you have ribosomes attached to it, proteins are going to be made there, and then they're going to be released from the cell. So let me give you an example of where that happens. You make enzymes, digestive proteins in your pancreas. But food never goes to your pancreas. Food never enters your pancreas. So where do those digestive enzymes do? They got to be moved. So they're made in the pancreatic cells, in the rough ER of the pancreas cells, and then they're shipped outside of the cell, and they're released into the small intestines. Okay? So what do? Well, they generally release them in these little bags little membrane bags called vesicles. So here's another picture showing you. See these little bags floating away from this rough ER area? These are little vesicles, and inside of these vesicles are those proteins or those enzymes that are made. These could eventually be released outside of the cell and make their way to your small intestines. Okay. Also, in the rough ER, this is where phospholipids are made. So phospholipids, in case you forgot, these are those fancy lipids that are part of the cell membrane. So the plasma membranes, or membranes in general, any membrane, would be produced here in the rough ER. Okay? Now, smooth ER. This is smooth. This is rough. They run into each other. Sometimes they connect with each other. It's okay. Just how it works. Now, smooth ER. Now, these areas of ER do not have ribosomes attached to them, so they are not producing proteins or enzymes. Okay, smooth ER is producing other things. It's producing lipids, hormones, and steroids that can be used. Steroids are used to make hormones, basically, that can be moved around. So these would be, for example, a lipid getting moved around in a vesicle. So they're also shipped in these little membrane bags called vesicles. All right. What else is a smooth ER? Well, you have a lot of smooth ER in your muscle cells. Why? Calcium ions are required for muscle cells to contract and to relax. So without calcium ions, you're going to have some issues. So they release and store calcium for muscle contraction. Okay. You also have a lot of smooth ER in your liver cells because they can process drugs like alcohol, for example, and potentially harmful substances. Your liver is loaded with smooth ER so that it can basically purify your blood of some of these toxins. So smooth ER, no ribosomes. Rough ER, ribosomes are attached. So let's look at this picture because it's going to lead into our next organelle. You'll notice that when these bags or these vesicles are made, they make their way to this other structure called the Golgi. I believe that's our next structure. Okay, so what happens is these vesicles, these little bags, pinched off, and they make their way to the Golgi. What is the job of the Golgi? The Golgi, I like to say it's the FedEx or the post office of the cell. What does it do? It modifies, it packages, it ships things around for the cell. What does it ship or move around? I don't know, hormones or lipids, proteins or enzymes that might be needed. So what happens is these vesicles, these little membrane bags, show up to the Golgi. The Golgi, they move through the membranes of the Golgi, and as they're moving through, these modifications get taken care of. Proteins get labels, meaning they get areas that they're shipped to specifically. So that's why I like to call it the FedEx or the post office. So they receive the vesicles, these membrane bags, and they ship things off in vesicle membrane bags. And this is, for example, a vesicle attaching to the plasma membrane, and they're releasing its contents. This could be some enzymes that were produced in the pancreas that are now going to be eventually released into the small intestines for digestion. Okay, what is it made out of? Just a bunch of flattened stacks of membranes, basically. That's what they're made out of. By the way, the Golgi is sometimes called the Golgi apparatus or the Golgi body. I generally just say Golgi to make it easier. Okay, lysosomes. These are special vesicles, so to speak. Why do I say they're special vesicles? Because they are membrane bags, which are similar to the way a vesicle is made. These look a lot like these guys over here. Okay, well, what's special about them? They have enzymes inside of these bags. They're called lysosomes. These are digestive enzymes, so they break down food particles. Okay, they break things down, and when they break them down, they can recycle them or remove them. They could be problematic. You could have some older parts, some older organelles that need to be broken down so that their parts can be reused to build up new organelles that would work. So they're made out of digestive enzymes, and they have a membrane around them. Okay, in humans, they're also used to destroy bacteria. Right? They have a very low pH. Well, not very. It's a 4.5. It's below 7. They have an acidic pH, which helps dissolve and start doing some of the helps the enzyme function in this case. So these enzymes are designed to function at a pH of around 4.5. All right. What happens? Where are they produced? Well, they're usually produced. Where do lysosomes get made? Well, the enzymes and the membrane are produced by the RER, and they get processed in the Golgi, and then they become a lysosome, there's pretty much a specialized vesicle that has digestive enzymes inside of them. Animal cells have them, plant cells do not have them. Peroxisomes, these are not on your main picture because if we were to put these on your picture, they would look a lot like lysosomes, and then they would create some confusion. They also are a membrane bag containing enzymes. Go figure. They would look exactly the same if we were to put them on your picture, so we are not going to have them on your picture. What do? They detoxify things like alcohol. So once again, you're going to have peroxisomes in your liver also because your liver and your kidneys are designed to purify things from your body. They help break down fatty acids. Okay. Fatty acids are not necessarily easy to break down. They're difficult to break down. There could be very long chains that are not easy to break down. They neutralize free radicals. What are free radicals? Well, free radicals are Oxygen ions, they could damage cells. They're linked to aging. It is believed that aging is connected to these free radicals that are produced as a result of these reactions we have inside of our body. We haven't learned this yet, but in our next chapter, we're going to learn about these oxidation and reduction type of reactions that takes place. When these reactions take place, free radicals are produced. And these free radicals, they're not so good for us. We need to remove them. Certain vitamins that we produce naturally in our bodies, certain vitamins we eat in our diets can help do some of this neutralizing for us. So these free radicals are associated with aging and other things of that sort. So peroxisomes have several jobs, okay? Several jobs, and they're very similar to lysosomes. The cytoskeleton, well, it used to be believed that the inside of a cell was this gelatinous material, liquidy, jelly-like stuff, and things were just kind of floating around. What scientists have discovered that's it's a little bit more coordinated. It is a little bit more organized than we ever imagined, but a lot more organized than we ever imagined. Inside of a cell is like a framework. So imagine this being a cell membrane and this being another cell membrane. This is the inside of the cell. These organelles, some of them are anchored and connected to these different parts of the cell. They're called microtubules, microfilaments, and intermediate filaments. Okay, intermediate filaments right here. Look at these guys right here. All right, what's their job? Well, they're like these protein cables or fibers. What do? Well, they reinforce this cell shape, and they anchor organelles in place. So they're attached and anchoring organelles in place. Okay, so these fibers are attached to everything. Microfilaments, all right, microfilaments. Oh, they're similar. They reinforce cell shape. They support cell shape. So there's some overlap. Beware of that. They're involved in cell movement. Okay, so let's find those. So right here, the microfilaments inside of the cell, there's these green ones, all right? All right, they're bead-like proteins, all right? And what do? They allow things to move inside of the cell. So things inside the cell need to move from point A to point B. They're involved in that movement, all right? So there's some overlap. Cell shape, anchoring organelles over here, involved in cell movement, moving things inside of it. Now, microtubules, all right, those are these guys. These are hollow tubes also, made out of what's called tubulin. What do these do? Well, oh, look, more overlap. Hold organelles in place, anchor organelles. Shape the cell, support the cell shape, support the cell shape. They're involved in that also. Act as tracks to guide organelles as they move within the cell. Oh, involved in movement. So there's overlap and differences. Beware of this on the test. You could find this difficult if you're not sure. So there's similarities, yet some small differences. They are all made out of protein, which are made out of amino acids. All these structures. So think of the skeleton framework inside of a cell is made out of protein, structure. Cilia, little hair-like structures. We have them in our lungs. Why do we have them down our bronchi, basically? What they do is if we inhale something, it gets caught in mucus, and these cilia can sweep it up towards our esophagus all the way back up, or we would swallow it back down to our stomach, and our stomach acids can destroy it. Right? So what are they involved in? Moving things. Fancy word, locomotion. And in our case, they're not moving the cell, they're moving things outside of the cell. Okay? They're found in eukaryotic animal cells. I just mentioned that we have them in ours, and they're found in these pond critters, these parameciums. They have them on their outside of their bodies, and they can use them to kind of swim around in the water. Right? They have a structure. It's called a 9x2 structure. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, and you have 2 in the middle. It's just structurally how they're made, and they're generally always made this way, the ones you need to know about. So they have 9 microtubule droplets, I told you, on the outside, and a pair on the inside. It's a kind of a structured feature that you always notice in them. They're almost like a propeller where they spin, too. Flagella, well, these are pretty much like a propeller. So this is a bacterial cell, and this is the flagella. These little hair things, these are not cilia. These are called pili. These are how bacteria can communicate with their environment and with other bacteria around them. So these are used for them to communicate. These are not for them to swim around. This flagella, this is like the propeller. It spins, as it shows you. They can swim in the water, all right? So once again, they're used for movement, locomotion of the entire cell. It's a whip-like tail, basically. They're found in bacteria, prokaryotes. They're found in eukaryotic animal cells. They're in sperm cells. We have them in sperm. That's how sperm swims to the egg. Some plant cells have them in their sperm also. Plants that specifically are reproduction for them is dependent on an aquatic environment will have that. And once again, they have that same 9x2 pattern of microtubules. So flagella and cilia are made out of microtubules, which are those proteins which are similarly structured in the cytoskeleton. Centrioles, oh look, they're also made out of microtubules. And what do? They aid in cell division. They're involved in cell division. We told you earlier that microtubules are involved in movement of organelles. Well, these are involved in moving the chromosomes around during cell division. Plants do not have centrioles. They're only found in animals. Cell wall. Now, you got to think of a plant. Plants can stand upright. All right. Two things are aiding. They need a lot of water inside of their central vacuole. They need their vacuoles full. If their vacuoles are full, they push against the cell and they allow the cell to stand upright. They also have cell wall, which is cellulose, which is fibrous material that can form the trunk of a tree and allow them to stand upright. So you got to think of the cell wall. This is like the bones of a plant. Plants don't have bones. We have bones. Our bones support and protect us. They protect our organs. They protect us. Plants, their protection and their support comes from their cell wall. Their cell wall, so they can't just have these giant fibers around them. They need water. They need pores. They need water and ions and molecules to enter and exit. So there's little holes in the cell wall called plasmodesmata. This area here is the central vacuole. Notice how big it is. It's pushing against the cytoplasm, against the cell. It's pushing the cytoplasm against the plasma membrane, which is pushing against the cell wall, basically. And this is what keeps the plant upright, basically. All right, this is how they do it. You got to fill the vacuole needs to be full of water for it to stand upright, generally, unless it's a tree trunk. But fibrous material will keep it upright. Plants have two parts to their cell wall. They have a primary and a secondary. The primary is on the outside. Okay, the primary cell wall is produced when the cell is still growing. Okay, when the secondary cell wall, are produced. The secondary cell wall is produced after the cell stops growing. All right. So this kind of creates a fibrous material, all right? So primary cell wall, while it's growing, this wall, while it stops growing, okay? Secondary cell wall. So they have a primary and they have a secondary cell wall. And by the way, they're not only found in plants. Students always think only plants have cell walls. Well, algae are in the protists. They're the seaweeds of the ocean. They have cell walls. Bacteria have a different structured cell wall. It's not made out of cellulose, okay? And fungi have a cell wall also, all right? So they have cell walls are not only found in plants, which what most students think. They're found in fungi, algae, and some bacteria also. The vacuole, this is the storage tank of a cell. In a plant, it mainly is holding a lot of water and helps the cell, the plant or the tree stand upright. So this is what it looks like when the vacuole is full. The vacuole is empty, you still see, this is the cell wall. When the vacuole empties out, the cell wall actually gets ripped away from the cell wall and the plasma membrane get ripped away from each other. So this can actually kill a plant cell also. When it runs out of water and it's losing too much water from its vacuole. So it rips the cell wall from the membrane and it will kill the plant basically that way. When it's full of water, it's pushing on all the edges, the plant will stand upright. We call that turgor pressure. Okay, so that central vacuole in a plant is called a tonoplast. Animal cells, we have vacuoles, but they're not that big. And the vacuole is surrounded by a membrane, so you don't have these in bacteria. You have these in eukaryotes. Chloroplasts. These are the locations for photosynthesis. And what's photosynthesis? The process of turning light energy into organic stuff like carbohydrates. Two internal compartments, okay, two phospholipid bilayers, outer membrane, inner membrane, okay. They have these parts inside of them. You have thylakoids, they look like poker chips, stacked, a bunch of thylakoids stacked, they call a granum. Stroma, which is kind of like the cytoplasm inside of the chloroplast, okay. By the way, chloroplasts, like mitochondria, contain their own DNA. They have their own ribosomes and enzymes. So chloroplasts and mitochondria have their own DNA, their own ribosomes, and their own enzymes. And chloroplasts are found in plants and algae, not just plants. Chromoplasts, these are a special type of plastid in the plastid family. What do they contain? Special pigments that add color to flowers and to the parts of the plant. They're involved in photosynthesis, but they're not the main pigments involved in photosynthesis. They're accessory pigments involved in photosynthesis. And they're found in plant cells. Finally, let's end with endosymbiosis. Now, there's a hypothesis. When I was in college, they called it the theory, but I don't know if they're still calling it a theory today because it's pretty hard to prove it. It's a great idea. It's a great hypothesis. So here's what it is. And like I said, they called it the endosymbiotic theory when I was in college. And now later on, they've refrained from using the term theory because we can't really support it with perfect data. But we have some nice supporting evidence as to why it's a solid hypothesis. Here's the hypothesis. It is believed that mitochondria, this is single mitochondria, and chloroplasts basically were formerly small bacteria or prokaryotes. It is believed that a larger cell engulfed or swallowed up these smaller bacteria. Okay, some of those smaller bacteria became the mitochondria. Some of those smaller bacteria became chloroplasts, for example. Okay, it's a weird idea. You're like, what? Really? Can it happen? Well, let's look for the evidence. Here's the evidence. Mitochondria and chloroplasts, I already mentioned you. They have their own DNA. And do bacteria have their own DNA? Yep. Do bacteria have their own ribosomes? Yep, they do. Okay. The structure of both the DNA and the ribosomes are very similar to bacteria. Oh, go figure. They both have similarly structured DNA and ribosomes, protein factories, and information. Okay. Mitochondria and chloroplasts, they can copy themselves and replicate themselves. So if you start working out a lot and your muscles need to make more energy, your muscle cells will actually duplicate their mitochondria. They'll make more mitochondria. You'll have more mitochondria in those cells over time. Okay. Also, mitochondria and chloroplasts are surrounded by a double membrane. All right. Oh, look, just like bacteria. And they have a similar similar inner parts as plasma membrane of prokaryotes. So the membranes are similar. The mitochondria and chloroplasts replicate like them. They have DNA, they have ribosomes, and they have their own enzymes. And all these things give us evidence as to that these mitochondria here and these chloroplasts were engulfed and became living inside of another thing. So symbiosis, living together, endo, engulfing them. All right. We will stop our lecture there.